to today's Tuesday lunch seminar. So it's my pleasure to introduce today's speaker, Dr. James Boyko. So James has come to us from, most recently from the University of Arkansas, where he finished his PhD just last August. And James has a bachelor's degree and a master's degree from the University of Toronto, where for his master's in particular, he worked on a particularly interesting groups of organisms known as squamates. He worked with Luke Mahler on some aspects of phenotypic evolution and evolution of sexual dimorphism in anoles. Somehow he ran, kicking and screaming away from squamates into comparative methods and even some plant things for his PhD with Jeremy Boyko at the University of Arkansas. And most recently here, he is now one of the new prestigious Schmidt AI and Science postdocs, this big sort of university-wide thing that we have going here now at the University of Michigan. So he has just begun a new postdoc, starting with that program, working on a bunch of really cool applications of machine learning to problems in comparative biology. And I've been familiar with James' work for a while. He has done a lot of very innovative work on issues in discrete character evolution, and he's an expert on some pernicious statistical problems that we in the field have been wrestling with for a while on, involving things like false positives have been plaguing a large fraction of the published work in this area. These are things that he's done. He's done a lot of work to resolve and I'm about to point the way forward on. And so with that, I'll turn it over to James to tell us something about frameworks for correlated character evolution and phylogenetic comparative modeling.
Thank you. So the mic's working? Cool. So this is going to be a pretty methods-heavy talk, and I wanted to give a little bit of background before I get into the methods about how I got involved in comparative methods, because I didn't start as a methods person. Close this. Okay, so like Dan mentioned, I did my bachelor's and master's at the University of Toronto studying the effect of congeners, so species within the same genera, affecting trait evolution in Lesser Antillean anoles. And the goal was to look actually at character displacement, so this idea that two species competing for the same resource are going to start diverging phenotypically, to try to understand perhaps the more well-known pattern of the Greater Antillean anole adaptive radiation, and the thought being that maybe character displacement was an early step towards driving phenotypic divergence in all of these various phenotypes that you see. And to sort of start gathering data, I went to various museums, would request specimens, and then take a series of different phenotypic measurements of these lizards. So I might take snout vent length, or head length, limb length, pelvic width, lamella width, all with the idea of testing my evolutionary hypothesis about how these extant forms were generated such that I could apply it to the Greater Antillean anoles. And I sort of, after I had collected my data, which is a large chunk of the project, I sort of ended up finally modeling it. And I would read in my data and run some phylogenetic models, which I sort of knew enough to know if I was testing my hypothesis or not. And I got some answer. I was like, okay, true. I'm satisfied. I know now what's going on. And I kind of got interested in this part of the pipeline, which is like, okay, I've taken all of these carefully observed lizards that I've measured with 0.01 precision calipers. My hands are pruny from collecting this data. Turn those lizards into numbers, which gave me a different number, and now that's my answer to the hypothesis. So like, what happened here? And for my field, this middle section is the phylogenetic comparative method, where we take our phenotypic data, combine it with evolutionary theory, and we test ideas of how the extant diversity was generated. And I really wanted to get a deeper knowledge of these methods. And I thought, okay, so for my PhD, where do I want to go? I want to learn these methods. Where's the best place to study this? Well, as Dan mentioned, it's obviously the University of Arkansas. Charles Darwin is known to possibly say that that is the place to learn comparative methods. And so while I was in Arkansas, I was working with Jeremy Boyko to study these methods. And my work in PCMs, which is phylogenetic comparative methods, has been focused on sort of two main themes, which is rate heterogeneity and correlated evolution. And so just briefly, when I say rate heterogeneity, I mean it like a very broad macroevolutionary scale, which is to say that if I'm modeling the evolution of some phenotype, there's going to be regions of the tree where things are evolving at different rates. So some parts of the tree might have a lot of transitions, some might have few. So not all things are going to evolve in the same way. And then correlated evolution is just that different phenotypes are going to, so change in a phenotype is going to depend on the state of another part. So evolution of different floral parts might depend. So the evolution of the gynoecium might depend on the current state of the andrecium. It's not going to be independently modeled. And in truth, most of what I'm going to talk about today has a lot to do with both of these topics. And in part, it's because the way that we model correlated evolution is sort of implicitly dependent on how we think about rate heterogeneity within the models. So let's start with this first part, which is the generalized hidden Markov model for discrete character evolution. So like I mentioned, the way that we test these hypotheses in phylogenetic comparative methods is to take our morphology, have a hypothesis, and see if that data matches, or if the model has a good likelihood for the data. And it requires two basic elements. The first is a repeatable measurement of form, and then we need a hypothesis about how that form changes through time. For discrete character evolution, probably the simplest model that we have is this Markov model. So we have two forms here, aquatic or terrestrial. I know these are very broad categorizations, but it is what it is. And then we have two transition rates. So we have a transition rate that you can go from aquatic to terrestrial, and a transition for terrestrial to an aquatic. And so for a phylogeny like the one on the left, maybe it's 15 million years old, 20, 30 species, having a single rate of transition, if you have multiple instances of aquatic and terrestrial, might be fairly reasonable. It might be at least an okay approximation of the process generating the diversity. Where we could get into trouble is if we're starting to look at very large phylogeny. So here is a phylogeny of Viridiplantae. This has a date well over 2 billion years, encompasses hundreds of thousands of species, and people have, and probably still will, apply these sort of simple Markov models where you have a single transition rate to explain the diversity of this. And it's sort of like, if you stop and think about it, it's kind of a crazy assumption to make that the way that rhodophytes are going to evolve, or mosses, or cyanobacteria, even if you're doing a plastid tree, is not going to be the same. Right? There's going to be rate heterogeneity, and there's going to be different ways that, even in this broad strokes way of modeling, we're transitioning between aquatic and terrestrial. It's going to be different throughout the tree. And so there's two main ways that we can incorporate sort of complexity and alternative rates of transition in our models. One is to account for some known factors that are influencing the transition rates. This would be something like correlated evolution. And the other would be to try to just infer where the evolutionary process is different. And so it's sort of like admitting our ignorance of the process. There's going to be places in this phylogeny where these transitions are not as simple as a single rate or two rates. And that data is going to exist in the distribution of the tips. And so rather than saying a particular character correlates with that, we're just going to say that we know there's variation, and we're going to try to model that variation. So the known factors, which is the correlated model, say I'm interested in aquatic and terrestrial angiosperms, and so I'm like, okay, perennial and annual life history, I think that's going to have an influence. I might suspect that if you are a perennial lineage, the probability that you transition to terrestrial is lower than if you were an annual lineage. Okay, so that's one known factor. There could be any number of factors. It's also possible just to say we don't know what they are. I know that there's going to be variation, and this would be the hidden model. So previous implementations, and so this unknown factor is what the hidden Markov model is really doing. So the previous implementations of hidden Markov models, and to an extent the correlated models, have focused just on this sort of binary system. So we have aquatic and terrestrial transitioning, or we have perennial and annual, and maybe you can model the correlated evolution. And most of the easy implementations of that, or the useful implementations of that, have been limited right here. So if you wanted to say, well, maybe aquatic isn't the best, and I want to differentiate between marine and freshwater on top of that, you wouldn't have been able to model that. Or maybe there's another category that you're interested in, another character. Maybe it's herbaceous and woody habit. You wouldn't have been able to model that. And then, of course, you can also extend this and have alternative rate classes. And so you can model this complex correlated evolution as well as alongside hidden factors. And so that was the goal of my first chapter of the PhD, was to just extend it. And most of like the likelihood and stuff is pretty easy to extend beyond what we had already done. So it was really just a lot of bookkeeping and generalization of the matrices. And I just want to highlight that, like, I have a series of vignettes, and you can use this starting with very simple implementations to teach you how to create your own and then biologically informed models. And rather than sort of discussing how the models are structured, I thought maybe an example to demonstrate how correlated evolution can be important would be pertinent. So here was a paper from Herbsakat et al. In 2017 that was a fairly big deal in the world of flower evolution, which purported to reconstruct the ancestral angiosperm flower. And one of the more controversial results was this finding of the shape of the flower, so the perianthendricium and gynoecium. And specifically what they found was a mix of whorled and then a spiral gynoecium. And someone who studied this replied to the paper and said it didn't make sense based on what they knew about the development of the flower that it would be a mixture of these two things. It should be either entirely a spiral or entirely a whorl. And so this paper marked the beginning of World War III. I'm pleased with that one. Okay, so to the author's credit, they were somewhat conservative about interpreting the ancestral states reconstructions, as you should be. But they also said that there was no comparative method that exists yet to account for the potential correlation of more than two discrete characters, which was true at the time. It's not true after the extensions, so let's check out what happens when I model the three binary characters with hidden states, although the hidden states aren't shown, in a correlated way. Well, I can take their data set, fit the model, and what I find is an entirely whorled flower, which differs from their original findings. So it's cool. It kind of means that a more realistic model of evolutionary change led to a more realistic result. So rather than modeling the floral parts independently, if you model them in a correlated fashion, you get a more realistic result. That's great. But it depends on if we can trust that these characters are indeed correlated, and if we can trust correlation in a comparative setting, which, based on the title of my next section, you might suspect is not a given. So we've all seen something like this. These are spurious correlations. In this case, it is margarine consumed and then the divorce rate in Maine. So this is a spurious correlation. There's no reason that the amount of margarine consumed is going to, well, no obvious reason that it's going to decrease or increase the divorce rate in Maine. And so it was kind of a shock to the comparative methods world when Madison and Fitzjohn published this paper suggesting that actually we very frequently find spurious correlations in these categorical character evolution models where we're looking at correlation. So I'm going to walk you through, these are 4 scenarios which they use to sort of demonstrate their point. And the left hand of the phylogeny is some character X evolving state 0 or state 1. And on the right hand is character Y. It's a mirrored phylogeny. It's the same phylogeny. And again, you have character 0 and character 1 for state Y. And so the question becomes, when you look at these these 4 scenarios, in which of these scenarios do we expect a true signal of correlation? So we don't want that spurious correlation. So on these left hand, we have replicated instances of one character arising followed by the other character arising. This might be more significant evidence of correlation, which is what the authors argued. On this side, we really only have one origin of the character X, and then we have some sort of unreplicated burst here of character Y, but it still goes down to a single origin. And so you can imagine this being the correlation of mammalian fur and the inner ear bone. There's not necessarily any reason that these two things should be coupled. They're just sort of 2 synapomorphies for the group. And so this, they argued, was evidence of a spurious correlation, and they called it phylogenetic pseudoreplication. But is this significant? So these left-hand ones are definitely significant. What if we were to test these scenarios? That's what they did. And they found, here are the p-values for likelihood ratio tests between an independent and a correlated model. They found in both situations pretty constant evidence for correlation, and in particular in Darwin's scenario, which is those two synaptomorphies being correlated. So this is problematic. Comparative methods has been using this sort of standard test for 30 years, essentially since Pagel came up with it. And so it calls into question those results. Other people agree that this is cause for concern. And previous attempts to sort of resolve this have focused much more on the careful consideration of your data set, which you should always consider your data set carefully, and the careful consideration of how you're going to model it. Both of these things are true, but it's somewhat unsatisfying because like no matter how much methodological vigilance you have, there's always the possibility that you're going to that there's going to be some signal in your data that's sneaking in based on that spurious correlation. So my contention is that this issue of false dependence was actually a failure to account for rate heterogeneity, and specifically rate heterogeneity not due to some other background focal character. So to get into it, I think it's important to talk more specifically about how we model it. So this is a Q matrix. This describes the transitions that are possible, the transition rates, the parameters of the correlated and the independent model. This is probably a lot. The way you read it is you go from the row, so x0, y0, to the column, so x0, y1. So this represents a transition in state y from 0 to 1. To make this a little bit easier, I'm going to just focus on one of the transitions, which is x0 to x1. And so in the case of the independent model on the left here, these colors represent the number of unique parameters. So the way that transitions occur here for x0 to x1 is the same regardless of the state of y. That's why this is an independent model. On the other hand, the rate of x0 to xy in the correlated model depends directly on the state of y. So you have if you're in the background state of y0, the transition might be faster or slower than if you're in the background state of y1. So that's the difference between the independent and the correlated model. If I try to replicate the Madison-Fitzgerald problem, I'm able to do so, and so that's what's shown here. This is using an evidence ratio, but essentially, the higher up you are, the more support for correlated evolution, and this is the classic comparison of a standard independent model against a correlated model. So we see pretty substantial support for correlation. So how can we account, if I'm right that it is rate heterogeneity sort of driving this result, how do we account for rate heterogeneity without having this character dependence, where it doesn't depend on the background state of y? Well, you expand it out into a hidden Markov model. And this kind of differs how people usually talk about hidden Markov models. Usually it's associated with some sort of hidden factor that we don't understand. But I'll propose like a slightly different viewpoint of it, that it's really just a Markov process that decides which process is going to be at play here. And so you have a Markov process bouncing between one independent matrix that represents this, and then it bounces also between that independent matrix. So both individually are models of independent evolution. We just have two ways of undergoing independent evolution. So if we take the original comparison, we have strong evidence of correlation, but if we add in this new hidden independent Markov model, we see weak, if any, evidence of correlation. That's great. That's sort of what we wanted. We didn't think the Darwin scenario should be attributing to correlation. Why is this the case? So let's again focus on this transition to x0 to x1. And you can kind of view this in a more rate heterogeneous way. So in this blue area, we have x0 to x1 very likely, right? There's no transitions backwards. You immediately, even if the transition didn't occur right there, it could have happened anywhere there. But the point is that you have this severe bias going to x1. And then in these white areas, or in these red areas, the x0 to x1 transition is very unlikely. So in our classic comparison, we see that which of these has rate heterogeneity at all? Well, it's only in the correlated model. The independent model has no way to account for multiple ways of transitioning from x0 to x1. It's only available in the correlated model. So when we expand it out using independent rate categories, we can find that the same sort of rate heterogeneity is allowed in the independent model, and the support for it decreases. And this was true across a variety of different scenarios that we tested that I'm not going to describe the difference of, but it was a fairly consistent result. Okay. So to this point, I have been discussing models of discrete character evolution, but there's a whole swath of questions and a whole bunch of characters that aren't really well described with discrete characters. So what if instead of perennial, annual, life history, or herbaceous and woody habit, I was more interested in generation time, or if I was interested in the correlation between these discrete characters and range size. Then I need a completely different way to, model these characters, because I'm not going to be looking at transitions between a continuous variable. So this last section before I get into the artificial intelligence stuff, is about that. So the types of questions that I'm sort of aiming to understand here are things like, does diet correlate with body size in mammals? So if you're a herbivore, are you larger or smaller than if you were a carnivore? Can a change in plant habit alter genome evolution? So perhaps annual or perennial life history or woody or herbaceousness influences the rate at which the genome, the size of the genome evolves. Or how does seed dispersal influence the climatic niche? So if you are a biotically dispersed seeds, are you in a more conserved niche because you follow alongside where the animals are dispersing to? And the way, sort of the classic approach that we've been taking is to use an Ornstein-Uhlenbeck process, which the change in your continuous trait is going to be described by this. So the change in form is going to be proportional to the distance between the current phenotype and some optimum, which is going to be inferred. So there's some optimal phenotype long-term, and the distance between those is important. And then it's scaled by this alpha parameter, which is sometimes called the pull of selection. It doesn't really have anything to do with selection in a microevolutionary sense. And then some additional random wiggle. So we can alter these parameters. We can increase the wiggle. We can increase the pull. We can change the optimum. And that's great. But if we want to sort of make inferences, we're at some point going to need to do this. This is a painting of a phylogeny in which we are going to say these are parts of the trees where some discrete regime, maybe carnivore or herbivore, are present. So we say that this is all, this is going to follow a theta for carnivore, and this is going to follow a theta for herbivore. The problem is that there are many different possible regime paintings. So you can, and we've never observed this process directly. We don't know if this was all carnivore, this was herbivore. We can make educated guesses or model it, but we don't know for certain. So there are two options for sort of incorporating that. One is to fit many possible regimes without knowing the discrete data. So these are sort of shift detection methods where we will paint and iterate through different possible mappings, see how that maps to our continuous character, and that will give us support or not. And so whichever one best suits the continuous character and the set of parameters, that's what we go forward with. The problem with this is that what if I'm interested in a regime painting that corresponds to a discrete character that might be evolving under those sort of Markovian processes I was describing earlier? So what if it is, if I want to say that herbivore and carnivore are transitioning between each other? That doesn't really affect these shift detection methods for the most part. The other option would be then to maybe I can fit my discrete character and then simulate some set of stochastic mappings and then fit my continuous character. So in this sort of system, I would fit my discrete data set, accounting for discrete character evolution directly. I would generate many possible stochastic mappings, optimize the continuous parameters for each of these mappings, and then summarize the result. The only issue here is that the continuous character doesn't really influence the generation of the mappings. It's based entirely off of the discrete fit. So it's a lot like the discrete trait is pulling the continuous trait. So the discrete trait kind of controls where the continuous trait is going to be evolving on the phylogeny. And so the argument for this new model was that previous iterations had focused on finding the single best mapping or not incorporating information. And I think that we should at least acknowledge that the history of these characters is highly uncertain, and that the discrete character, if it is something that has undergone evolution along the lineages, should also be accounted for. So the Howey pipeline tries to create some sort of feedback between the optimization of the continuous parameters and the optimization of the discrete parameters such that they influence each other. And so it's more like this, where the continuous trait and the discrete trait are working together. So there are a lot of sort of technical challenges involved with developing this model. So I'm after the joint probability of a discrete and continuous character. The first stop is the probability of the continuous trait given the underlying regime. Thankfully, the math for this is worked out already. I'm going to be using the most generalized model, which is that of my PhD advisor, Jeremy Beaulieu, in which all of the different Howey parameters or OU parameters can vary. The only issue with these approaches is that they had relied on matrix inversion, which for large trees is a very slow process. So I implemented this linear time algorithm. It essentially just makes the calculation much faster. And I'm going to need it to be faster because I'm going to be evaluating over several different discrete mappings. The next thing I needed to do was get the probability of the discrete regime and trait. And so I had tried, and this is going to be following the hidden Markov models that I've described before. And I had tried using a probability density in which I was evaluating the sort of continuous mapping, but it was very, giving me very odd results. So I defaulted into doing a probability mass in which I'm only looking at nodes and then sort of assuming a painting throughout the branches otherwise. The last thing that I needed to do was to generate the mappings. And that's easy enough. We have algorithms to generate stochastic maps based off of a discrete character or a discrete model. The problem is that when you try and fit this sort of equation to it, most of the maps are really bad. So they don't contribute very much to the overall likelihood. It's as if we took our average incomes. It would be very different if it was us and then Bill Gates walked into the room and then we added his income in here, right? The ones that are contributing to the overall mean are occurring in this section. And that is what's going to be driving the overall likelihood for a given set of parameters. So it's really about finding this best set of mappings that was important. And so to sort of try to get at that, I'm not going to go over it in detail, but I made an algorithm which tries to incorporate information from the continuous trait in an iterative process in which we assume the ancestral state is the theta, and then we assume it's the expectation of the best maps. It's a bit of a kludge, but it does seem to work. So we start finding, when we sample our mappings under this algorithm, higher joint likelihood mappings. So then the next step is to test the different models, and so it has all of those discrete parameters, it has all of the continuous parameters, and for the most part it performs fairly well. So this is just the difference between the true estimate and the generating estimate and the generating model parameters and the estimated model parameters. And the only place where I get into some trouble is these variable alpha models, and that's fine because well, it's not fine, but they have always given OU model problems. Okay, so the caveats, there are none. This model is perfect. No, it's not. It's far from perfect. It's just the first step, I think, to these joint discrete continuous models. So one thing that has always bothered me is that those discrete mappings that I'm talking about, they're not really treated the same way in the discrete and continuous process. The continuous sees them as it can be any sort of value between them, and the discrete just sees them as sort of node-wise. So it's not that one's a mass and one's a density. It's just the continuous doesn't see them the same way that the discrete does. The joint map generation, like I said, is a bit of a kludge. It'd be much better if there was like a true incorporation of both probabilities to generate the mappings. The stochastic map generation is slow. It makes it difficult to fit large data sets. It makes it difficult to fit complex models. And the variable alpha models remain difficult to estimate, and they've been difficult to estimate in previous OU models, and they still are with this. And the final thing I want to talk about is that this is a very parameter-rich model, and I want to spend some time to just discuss what that means. So this is the basic Howie model. This is a complex one, but we have 2 transition rates. We have 3 parameters with each. So for this basic model, maybe we're looking at 1 continuous character, 2 discrete characters, 8 parameters. If I want to add in another discrete character, and if this is one organism, it makes sense that we would. We're now looking at 28 parameters. If it's a large tree, maybe we need a hidden state, and now we're at 58 parameters. If I add another discrete state to each of these characters, we're at 110 parameters. And so we're just sort of falling in to fit more complex models, we need more data. But the more data we have, means we need more complex models. And the question is, which of these is going to grow faster? And although there's no explicit work looking at this in comparative methods, my intuition would be that the amount of parameters that we need is going to outpace the data that we can collect. So we need some sort of alternative, especially when we're starting to think about more complex data. And so that's where the final part of this talk comes in, which is applying artificial intelligence to ecology and evolutionary biology problems. So AI, machine learning, deep learning, I think we've all heard these terms, but I wanted to contextualize them because I'd never, prior to starting this postdoc, really taken the time to appreciate the different stages of this. So artificial intelligence is the effort to automate intellectual tasks performed by humans. Probably one of the best known examples is the chess-playing robot Deep Blue, which defeated Garry Kasparov in 1997. What makes this an artificial intelligence is that we have programmed the rules of chess alongside a set of just general what-if situations. So if Garry was to play e4, the robot might play e5. And so we have a bunch of what-if statements to create an outcome. Machine learning, which is a subset of artificial intelligence, is where we train the model to find the statistical structure in a set of examples, to produce rules for automating a specified task. I think it's well captured in this graphic here. So if the classical way that we've been talking about modeling or programming is to feed in data alongside a bunch of rules. So I have some model of how evolution might proceed. Those are the rules under which the data is going to be manipulated. And then we get some answers out, which might be the likelihood, might be some sort of hypothesis answer. With machine learning, the goal is different. The parameters don't have much meaning in the ML models, like I'll talk about, but we give it data, we give it answers, and out pops some rules. The tricky part, at least in evolutionary biology, is knowing what these answers are and sort of thinking about what that could even mean. You know, and sometimes when we're doing evolutionary biology, it's like, okay, if it's a more species-rich clade, we'll say it's more successful. But now, if I was to put this into a machine learning algorithm, and it's like, okay, here's a skull, is this more successful or not? It's like, no, you don't measure that. We sort of infer it from other things. So thinking about what these answers could be is one of the challenges I'll be facing during this postdoc. So the final thing I want to talk about, because deep learning is going to be important going forward, is deep learning is a subset of machine learning, and it learns successive representations to transform the input data into a meaningful output. So this is a classic example from handwritten digits. So we input this handwritten 4. The machine learning algorithm, it knows what the answer is for, but it trains itself to sort of learn representations that more easily guide it to answering the 4. Whether or not these representations are super meaningful is an active area of research. But the goal is to represent that original input in a way that it's very easy to recognize that that is a 4. Okay. So why apply deep learning to ecology and evolutionary biology? So deep learning might be our best option for highly complex multivariate traits. I showed you the example with the Howie model, where we had discrete characters, only a couple of discrete characters, maybe 6 states total, only one continuous variable, and we're already up to 110 parameters. We're going to need a massive tree and a huge data set to do that. What happens when you have a 3-dimensional mesh? How do you model that in sort of the comparative methods that I've been talking about, right? What if you were looking at an image of maybe butterfly coloration or snake coloration? It becomes very difficult to think about how we're going to model these complex traits and even how we would represent these complex traits. I think that it'd be very useful for data collection. I think it's always good to standardize and automate data collection. So this is an example of photogrammetry. This doesn't necessarily need to use artificial intelligence, although it can be improved with it. So the idea is that you take a bunch of two-dimensional images, you align them in such a way that you can represent it in a 3D mesh, which is really cool, because imagine going out into the field, taking a bunch of photos of your flower or specimen, and then being able to put it back, and then you have this incredibly complex 3-dimensional data set. It would make the data collection a lot faster. We can use species identification. I think there's a lot of potential to automate data collection with machine learning. And then the final thing I think is useful is that the current modeling that we do through comparative methods, I think restricts how we think about the questions that we can answer. So often, you come across a paper or proposal which is like, I am going to correlate character X with speciation rates. It's like, yeah, that's always worth doing, but I think that we're limited in obviously the models that we can run, but I don't think that we should be limiting how we think about the evolutionary process based just off of those models. We know that evolution isn't Markovian. We know that it's probably not following Brownian motion or OU models. So I think there's a potential for deep learning or machine learning in general to improve that and maybe expand the hypothesis or how we think about these questions. And I put the sloth here to slow down because it's about the point that deep learning is not also the end all, the be all end all, right? It has limitations. It's not the silver bullet that's going to solve all of our problems. And I think the biggest limitation to machine learning is when we think about how we have typically derived advancements in modeling. So usually we contextualize them or strictly derive them from some sort of biological understanding. We understand how the process goes, and so we try to find a model that matches that. This means that we have parameters which have very well-defined meaning, which is great. This is in contrast to the models of modern machine learning, where interpreting the parameters is very, very, very difficult, if not impossible in some cases. You don't directly interpret the parameters of your machine learning model. You have to sort of finagle it in such a way that you can get interesting representations of your data and sort of understand what's happening. So if we can't interpret parameters, what's the point of doing this? So I think it might be more important at this stage to, especially for this highly complex multivariate data, to discover meaningful representations of the data. So this is a classic example of Roth shell morphology, in which maybe distances between different points of the shell aren't as meaningful as the underlying parameters which might generate the shell. Or if we have this more computer science example of the Swiss roll problem, in which if we were to just squish this down and project it to a two-dimensional plane, we see just like a mess of points, but what we really want to do is unfold it, right? We want to take this two-dimensional plane and roll it outside of the 3 dimensions. So the idea here is that this really complex, this seemingly complex 3-dimensional roll is actually just a 2-dimensional plane. If we can represent it as such, we might understand, or might have a better grasp of sort of the evolution of the structure. If this is a better representation of the actual evolution. And so this was just my reminder that, imagine trying to use Brownian motion or Ornstein-Uhlenbeck models to simulate this creature. It's not going to happen. depending on how you do it. Imagine applying it to just each individual point. It's not a thing that can be done. And I want to just show a naive use case of machine learning in my own work. I only started about a month and a bit, 5 weeks ago, so it's all very preliminary. But this case study is meant to sort of act as a sanity check. So this is a variational autoencoder, and what this does, because I don't know how to label this, I'm not going to label it a lizard or a snake or a dog or a cat. I kind of want to understand the essential features of this. I want to be able to unroll, if this is the Swiss roll, I want to be able to unroll and sort of learn what are the important dimensions upon which it's evolving. And autoencoders learn to represent themselves, essentially. So you give it input data and it learns to represent that input data. So it has two sort of neural networks. It has an encoder, which takes this really high dimensional object and squishes it down into 1 or 2 dimensions, or a few dimensions. And then they have a decoder, in which we take that latent space and we decode it to recreate what we had put in. And sort of the hope is that within that latent space is the 2-dimensional plane of the Swiss roll. That is like the essential parts of the organism that is evolving. And it's because it's gone through this information bottleneck. So we've taken this really complex form, put it through a bottleneck, and then had it recreate that form from those simple dimensions. And this differs from sort of the typical morphometrics approach in which we're trying to conserve all of the information. In this case, we're explicitly not trying to do that. We just want the essential features. There's some evidence to think that maybe VAEs are a little bit better at parsing complex data sets. They spread out a bit more than PCAs. I wouldn't read too much into this because it's much easier to interpret a PCA and what those axes mean. The variational autoencoders is much more difficult to understand what these axes are doing. Okay, so here's my naive use of autoencoders. This isn't, I'm not going to go into in depth. This is a data set that Dan shared with me from Jacques Gauthier, looking at essentially trying to assemble a morphological tree of squamates. And you can obviously do this through classic parsimony type analyses. You can figure out sort of morphological distances and create a neighbor joining tree. And I applied this I just used this on an autoencoder, and I got kind of what I was expecting. I wanted to find sort of meaningful groupings of it. I don't know exactly how to interpret the parameters. I don't know how to interpret the axes, but I did find that snakes are weird, and they are weird. So that's great. It means that there is at least, it's pulling out something that is meaningful. What exactly these axes mean, I don't know, but it would be fun to sort of search as to what that means. But we know snakes are weird. It pulled out that snakes are weird. That's a good start. At least it's not an insane model. So, and I'll end by just saying, obviously, the machine learning techniques as applied to this sort of morphological data aren't super interesting when it's a bunch of binary characters. But what's interesting is that these techniques apply equally well to 3D meshes as they do to images, and there's all sorts of machine learning tools that are specific to the problems and to the data sets. So I think it's a very fruitful way forward for evolutionary biology, and I'm excited to dig into it more. So I'd like to thank all of these folks here. I'm really happy to be here at Michigan. It's been a lot of fun discussing with all of you, and I'm happy to take any questions.
So I have a bunch of questions. Like for how you have parameter explosions, so you need to regularize, right? You need to put some further structure on all this parameter stuff. People explode that.
Yeah, you can. Yeah, so we do, like, we'll do AIC to fit the models. Is that what you mean in terms of regularization?
No, I'm talking about more like an example would be assuming your parameters, if you organize them into a matrix, that matrix is low rank or something like that.
Oh.
So,
no one's explored that.
yeah, so that's probably, yeah, you're right that if you got more data, you'll need more parameters and they'll go up, but that's one thing you can
It's a good idea.
And for the autoencoders, so there's, I don't know how deeply you've got into it, but there are these people that are trying to learn latent tree-like representations of data from autoencoders.
Mhm.
Seems like a better fit for this than
Yeah, it would be.
Euclidean embedding. Have you looked at that at all?
I've come across network. What's it called? Graphical neural networks. Is that related to it at all?
I'm not sure.
No. Oh.
We can talk about that.
Yeah.
So let's take a well-defined parametric problem in evolutionary biology, like phylogenetic construction, where everybody's obsessed with parameters. You have great parameters. You have ancestral states. If you want to do something like infer phylogeny, so you're predicting in like 15 years, you think we're still going to care about parameters in that sense? Where in a sense, those are nuisance parameters with respect to our actual object of interest. For most, in many contexts,
Yeah.
you're going to care about these anymore in 10 or 15 years.
I don't know. It's hard to imagine because I've spent like 5 years caring about the parameters and thinking deeply about them. But yeah, the machine learning stuff is we don't care about the parameters of the model. We sort of care about the interpretations of it. Which I think it's interesting because it refocuses our efforts on like coming up with good questions and coming up with good data sets and thinking about how the biology actually works, rather than focusing on the parameter estimate. But I don't know. It depends. We'll see how useful it is. I think there's potential though that we will move beyond parameters. Yeah.
Question from Bruce Martin: how well developed are variational autoencoder algorithms in the case of continuous?
Pretty well. Yeah. They're definitely like if it's not a variational autoencoder exactly, there is something that will do the same job.
He said sweet.
Cool. Good.
How much of a so thinking about your second part of your talk on discrete and continuous traits together, how much of an improvement do you get from thinking about them jointly in the sense that you are versus thinking about them independently? So you are treating them completely decoupled, but you allow great heterogeneity, hidden states to deal with the discrete part of it, and you work from there with that set of mappings in the continuous trait state and on the continuous trait. Because what is your view in terms of obviously it makes sense to investigate that and ask the question, but now having done it, what is your assessment of what the actual gain is?
Yeah. So I think it depends on the things that you're correlating. So I did a study looking at climate evolution and life history across several different groups of angiosperms. And ancestral states, they're not like the best thing in the world, but it was interesting that if I reconstructed the annual, I could reconstruct the root state as either being annual or perennial, and depending on which climatic variable I chose, I could be very strongly confident that the root state was annual or very strongly confident that the root state was perennial. So there's clearly an effect. The strength of the effect is obviously going to depend on the data set, though, but it does change things.